Hello everyone, welcome to a new session on metabolism. In this session, we'll be dealing with the metabolism of proteins actually. Metabolism of proteins, it is nothing but the metabolism of amino acids itself, and that is why the heading has been given as the amino acid metabolism. Here what happens is the dietary protein, that is a protein which reaches the body through diet or even the protein in the body. Those get undergoes proteolysis by way of digestion in the gastrointestinal tract and produces the respective amino acids. And these amino acids, it may be taken through blood to various parts of the body. Especially in liver, various oxidation of amino acids takes place, like by the way of transamination, deamination, or decarboxylation. We can see that the amino acids, it can be converted into nitrogenous derivatives or nitrogenous compounds by way of various other processes. Now, what we are going to deal in this presentation is more about the oxidation methods, the transamination, deamination, and the what you call decarboxylation methods. Okay, it is catabolic degradation of amino acids we are more concerned with. Okay, now how the oxidation of amino acids are classified to. It is actually for convenience of understanding the oxidation of amino acids, or we can say the catabolic degradation of amino acids can be classified into three categories: the conversion of alpha amino group, breakdown and conversion of carbon framework, decarboxylation of alpha carboxyl group. Simple thing, alpha amino group is the amino group which is directly attached to the alpha carbon. Okay, I hope you remember there is a carbon to which asymmetric carbon to which four different groups are attached directly in the case of amino acids, that is amino group, carboxyl group, a hydrogen, and a side chain. And it is this side chain which decides the nature of the amino acids, right? The physical chemical properties of amino acids, it is decided by the side chain. Now the amino group which is directly attached to the alpha carbon is what is referred as the alpha amino group, and the carboxyl group directly attached to the alpha carbon, it is the alpha carboxyl group. So these two undergo changes, and that is what happens in the oxidation of amino acids. So oxidation of amino acids, it can be divided into conversion of amino group, the alpha amino group specifically. It could be of three kinds, or it could be done by three methods, we can say. Here you can see deamination, transamination, and transdeamination. So what is actually conversion of alpha amino group? The alpha amino group of the amino acid, it gets converted into ammonia. Okay, and this ammonia, in the case of deamination and transdeamination, this amino group, it gets converted into ammonia, and the free ammonia, it is liberated. While in the case of transamination, this liberation doesn't take place. So the transamination does not involve what you call the release of ammonia. Okay, it is just transfer of amino group from one to the other amino acid. Okay, now degradation of carbon framework, whatever is remaining, like even the carbon framework in the R group, the side chain, it undergoes degradation. Okay, and it happens when it enters the citric acid cycle. Okay, so that is what happens in the degradation or breakdown and conversion of the carbon framework. Okay, the third one is the decarboxylation. That is, as already mentioned, the alpha carboxyl group undergoes decarboxylation, right? And this is also a very important oxidative methods. Okay, we will see in detail. Now, conversion of amino group, that is the first one, right? Alpha amino group, and here it involves the cleavage of amino group, of alpha amino group, from the amino acid as ammonia, and it takes place in through three reactions, or we can say there are three types of reactions which involve the conversion of amino group: deamination, transamination, and transdeamination. Okay, what is deamination? Deamination is nothing but deamination is removal of amino group from the amino acids as free ammonia. Okay, and this results in the liberation of ammonia, which is used for urea synthesis. And we can say that the carbon skeleton of amino acids which are produced as a result of what you call removal, after the removal of ammonia, whatever the carbon skeleton of amino acid is there, it gets converted into keto acid. Okay, so we can see that the amino acid, it undergo deamination and the output is release of free ammonia plus the formation of corresponding keto acids, or we can say hydroxy acids. Okay, in vertebrates, the major site of deamination is a liver mitochondria. Okay, and there are four major, even in some cases, we can say some may take in even the kidney becomes a site for deamination. There are four major types of deamination: oxidative, reductive, hydrolytic, and intramolecular. Oxidative, as the name suggests, there is oxidation redox reaction taking place. Okay, reductive, removal of hydrogen. Hydrolytic involves the water molecule, and intramolecular deamination, that is, we can see here, it is within the molecule of the amino acid itself. There is no addition or what you call this one of it is removal of ammonia, and whatever is remaining, the carbon skeleton, it undergoes intramolecular what you call shift and produces a new molecule. Okay, we'll see it in detail here, and in detail we will study the oxidative and hydrolytic. Hydrolytic deamination is also referred as non-oxidative. So these are the two ones which we are going to study in detail. That is oxidative and the hydrolytic. Okay, but we'll see the other three. I mean, in general, what are these? Reduction deamination or reductive deamination. You can see this is the amino acid. This is the alpha carbon. This one is the alpha amino group, alpha carboxyl group. The hydrogen attached to the alpha carbon and the side chain. Okay, so in reductive deamination, what is going to happen? Reduction means addition of hydrogen, right? So the hydrogen ions are added, and what results it is fatty acid. Okay, so amino acid, it gets converted to fatty acid with the liberation of ammonia in the reductive deamination. Okay, the corresponding fatty acid. I hope it is clear, right? Hydrolytic deamination involves the addition of water. So this is the amino acid. Then we have water is added. What is produced? A hydroxy acid. Okay, hydroxy acid is produced and ammonia is liberated. Okay, in all the deamination process, ammonia is liberated. Free ammonia is liberated. Okay, so hydrolytic deamination involves addition of water and conversion of amino acid to hydroxy acid with release of ammonia molecule. Intramolecular deamination, here again amino acid here. Okay, so ammonia is released. Like when ammonia is released, what happens is it is NH3, right? So here what happens is there is a double bond formed. Okay, it is CH double bond CH. So it becomes unsaturated. It is a fatty acid because it is having a COH group at the end, and it is unsaturated because there is a double bond over here, CH double bond CH. Okay, so in the intramolecular deamination, an amino acid, it will result in the formation of unsaturated fatty acid on the removal of free ammonia. So in all the deamination, ammonia is the product and it is released, and you can see what are the products produced other than ammonia. It is apart from ammonia, we can see fatty acid, the hydroxy acid, and the unsaturated fatty acid in the intramolecular deamination. When we speak about oxidative deamination, it involves the use of NAD plus. As already mentioned, it is a oxidation, right? So here NAD plus is the oxidizing agent, and alpha amino group, or sorry, what you call amino acid, it gets converted to keto acid with the reduction of NAD plus to NADH. Okay, and ammonia is released. Okay, so it is alpha amino acid, it will get converted to keto acid, and during this time NAD, it gets converted to NADH, and ammonia, it is being liberated. Okay, so this is just a general idea about deamination. We'll be looking into oxidative deamination and hydrolytic deamination in detail in the coming presentations. Thank you.